afternoon, everyone. My name is Lin, and I'm a summer intern with Braggins Lab at Vanderbilt University. Today, I'm going to present my project, which is about color map for enhanced visualization for molecular image data. So let's first talk about the intention for this project. So as we know that HuBMAP is committed to developing an open and global platform to develop the map for healthy cells in the human body. If the platform is faced with different type of audience, then a graph that is simple and easy to understand will be a better way for people who use this platform. So we just find out that color can make it easier for people to find out the key point in the image and easier for them to distinguish the different part of cells and easier for them to find the information they want. And our project will focus on creating the highly complex molecular image with color in order for people who use it to better get information from the color map, such as getting the number of molecules, dynamic range, and the rate of molecule change by time. Our project is collaborate with Jonathan, who also work on the color map, but for some other different type of applications. We have applied the program from Jonathan to create the color map for IMS image. The goal for our project is to create the color map based on the luminance of the IMS spectrometry image in order to enhance visualization and get a better perception of dynamic change. Because we have to add the color one by one with different mass to charge ratio for the color map that we have right now, then for our project, we want to add the color automatically by the program with a appropriate color, which can show the information we want the image to show, so that we don't have to spend too much time to find the appropriate color for each IMS image and add them one by one. As we can see, there are two images here. The image on the left will be the color of the original IMS image, and the image on the right is the image with the color that add on based on the luminance from the image on the left. Different luminance will give different color to the image so that people can be easier to separate apart the cells and getting the information from the cell. Then what is the method or process that we have to do in order to get this kind of color map? So first we will get the IMS image from mass spectrometry, as we can see on the left. And then we have to use a program to give the image color. In this program, Matplotlib has played a very important role. As you can see, we have imported this into our program right here. Because it's a comprehensive library for creating static, animated, and interactive visualization in Python. In here, we are going to use it to create the static color map. And then we have several programs that use to gather luminance from the image and create the colorable color scale. And after all this thing, we will insert the IMS image and give its color based on the luminance that image has and display the image. Here is our result looks like. As we can see, there are 4 images with different color scale. The one on the bottom left, this one is the one we create the color scale for. And the other 3 has used the different given color scale from Matplotlib, which has shown right here. As we can see, there are many different types of given color scale for us to use with Matplotlib. But for the most important and most difficult things for our project is to create the color scale on our own based on the luminance. As we can see, the image with the color scale we create more clearly shows there are 2 different types of ion in this tissue than the other 3 images with different color scale. Because we make the color scale by our own, then we can be more easier to add different meaning to different colors. Then some idea just pop up, which is exactly to give different color, different meanings. The place we can usually see color map is with the weather forecast. With the weather forecast, we can see give different color, different meaning. For example, the color can tell you if it's a light rain or heavy rain today. We can also apply this to the color map of cells. For example, use it to determine if it's a normal or not normal health disease cells, or use it to determine the ion percentage in different places of cells, which we can make as same as the probability of precipitation. Also, we can see on the right, we can make some warning like this for disease cells. For example, how serious is this disease gonna affect these cells, or how this disease can affect human bodies. Also, colors have different intensity. They can also represent different meanings, such as dark colors can represent that this amount of certain ion in this area has a huge difference from the normal healthy cells and the disease cells. And light color can be used to represent a smaller difference between the healthy cells and the disease cells. With this summer internship, I have really learned a lot about how to do different experiments with different instruments, such as MODI IMS, which is the experiment that I have to do in order to get the IMS image. As we can see, there are 3 images here. The first 2 is the individual IMS image, and the third one is the overlap of IMS image. In order to get this kind of colorful image or colorful color map, we have to give the color to every individual IMS image and then overlap them together. Also, I have learned something about biology data analysis, especially from the project that I have done. I have learned how to create an environment for us to write a program, how to write a program to create color map using Python with the Jupyter notebook. Besides that, I really feel the importance of interdisciplinary learning and teamwork. It can really make the project work more efficiently because everyone knows what they can do and they understand what they are doing. And the biggest challenge that I have met is at the very beginning of this internship, I was so confused every day. I don't know what I have to do or what I can do because I'm not really familiar with the workflow in the lab. But practice makes perfect, and also because of everyone in our team's help, everything becomes easier right now, even though I have met some challenge while work on my project. And lastly, I would like to say thank you to Dr. Jeffrey Spraggins, his Patterson, Martin Defense, and all other members of the lab for showing me a lot of experiments that I haven't seen before and helped me to figure out the problem that I have met with my project. Thank you, Jonathan, for the color map program support. And thank you to Zarina, AJ, Maya, and Julia for their weekly support and guidance. And thank you to HuBMAP program and NIH for funding and give me this opportunity to learn more and be part of this. And thank you everyone for listening, and just let me know if you have any questions.